This is video 3 of chapter 4. This is our final video of material for exam 1 in anatomy and physiology. We're going to go over two sections 4.3 and 4.4 in this video. That means this video is going to be a bit longer than average, but these two topics are so closely related, it made sense to put them together. So we're going to start by looking at the cell cycle and how DNA replicates. And we'll talk about some errors in replication and also how that cell division is regulated. Then we're going to do a quick review of heredity. We're going to talk about how chromosomes are arranged, what alleles are, and how certain traits are inherited. So jumping into cell division now. Before a cell can physically divide, we have to replicate the DNA. So that way, each one of the daughter cells, the cells that are produced by the cell replication, have a complete copy of the DNA. This happens at the cellular level, and it also happens very quickly. So this has to be a very precise process. We use the law of base pairing, which we've talked about a lot before, where A and T pair together and C and G paired together, and that allows us to end up with identical daughter copies of the DNA. DNA replication happens in 4 steps. First, the DNA unwinds from those histone proteins that we've talked about previously. Then, an enzyme called DNA helicase, remember ASE tells us it's an enzyme, DNA helicase unzips the two sides of the DNA that breaks the bonds, the hydrogen bonds between the bases. The places where the DNA helicase begins to unzip the DNA are called replication forks, and this is where DNA replication begins. The next enzyme involved is called DNA polymerase, and DNA polymerase makes the new DNA strands. They add in base pairs to complement what is on the existing strand of DNA. And finally, the newly made DNA is repackaged. Enzymes wrap it around the histones, and the DNA takes its double helix form that we expect to see. So here's a visual representation. We have the parent DNA. We have the DNA helicase that is breaking apart the two strands. We have the replication fork here at the very edge between the parent DNA and the separated DNA. And then we have the DNA polymerase that is adding in the base pairs as the DNA strand grows. This is a very precise process. For every 1 billion base pairs that are added, we typically only have one error, and this is because DNA polymerase has several spell checking, for lack of a better word, features that double check and make sure mistakes aren't made, and when mistakes do get made, they're fixed immediately. On the other hand, we have something called mutations. Mutations don't happen due to replication errors. Mutations happen as a result of mutagens in the environment, such as radiation, think UV radiation from the sun, or viruses, think of the HPV virus, which can cause mutations in the cells of the cervix, and some chemicals. So some mutations have no impact. They don't impact the organism at all. Other mutations are lethal. They kill the cell no longer functions at all. Some cause cancer and some cause genetic defects in future generations. So now we're going to talk about the cell cycle. So the cell cycle has 2 broad phases. First is interphase, when the cell is growing, duplicating its DNA, and living, doing whatever that cell is specialized to do. And then we have the mitotic phase, where the cell divides. Interphase is divided into G1, G2, and G3. G1S and G2. And then the mitotic phase is subdivided into 4 parts: prophase, metaphase, anaphase, and telophase. We're going to go through these in detail. Okay, so our interphase is shown by this green portion here, and like we just talked about, it has G1, S, and G2. G1 is where the cell is doing whatever it's specialized to do, growing, dividing, whatever that cell does. S phase is where the DNA is replicated, and G2 is also a living, doing normal cell function phase, except the cell has double the normal amount of DNA in that phase. Mitosis, like we talked about, is where the cell divides: prophase, metaphase, anaphase, telophase, cytokinesis. So new information, G0. G0 wasn't on the chart we looked at previously. During G0, the cells are not doing normal metabolism. They're in a dormant or a resting phase. Some cells go into G0 for a short time. Some cells stay in G0 permanently. Examples of G0 are nerve cells that they can't divide, they can't replicate, and if they're damaged, they can't be replaced. So think of a spinal cord injury where once the spinal cord is severed, because the cell is in G0, it's not going to be regenerated. Also, unfertilized eggs. Humans, for instance, human females are born with all of the eggs they're ever going to have in their ovaries. So those unfertilized eggs that stay completely dormant until puberty and then mature one by one every 28 days, those cells are in G0 as they're resting there. Some cells go through the mitotic phase much more frequently than others. Think of skin cells, for instance, a cell that needs to be replicating quickly because it needs to replenish itself often. Those are things that are going to be going through mitosis very often. Other cells go through mitosis much less frequently. Think of an intestinal cell. An intestinal cell can last much longer than a skin cell. It doesn't need to be replaced as often. Talking through the 4 phases of mitosis, these are important, and what happens in each is important. The name is important: prophase, metaphase, anaphase, and telophase. Okay, the whole idea of mitosis is to produce 2 identical daughter cells, 2 offspring cells. So we do this by the process of mitosis. As our cell enters mitosis, the chromatin condenses, that DNA changes its form slightly, so the chromosomes become visible. From prophase to metaphase, some books call this prometaphase, this intermediate. The nuclear envelope, that double-walled nuclear envelope, breaks down. And then in metaphase, the chromosomes line up in the center of the cell. During anaphase, the chromosomes are pulled apart towards the poles of the cell. In telophase, the new nuclei begin to form, and the 2 daughter cells separate. So this is mitosis. The cell has some criteria for knowing when it should divide and when it should stop dividing. Okay, so cells divide when there's enough cytoplasm in the parent cell to accommodate 2 daughter cells. The cell divides after DNA replication happens. The cell divides only if there's enough nutrients around, and only if certain growth factors are available, and only if the cell senses it has enough space. Cells stop dividing when they're in contact with neighbors on all sides, when nutrients or growth factors are lacking, or when they're in close contact with all the other cells around them. In order to continue through mitosis, cells have several checkpoints that they stop at, and the cells go through a process where they make sure their DNA is intact in order to move on. So before S phase, there's a checkpoint, and before mitosis, there's a checkpoint. There's also a checkpoint between metaphase and anaphase. So at each of these points in the cell cycle, the cell stops and examines itself for any abnormalities before it continues to the next step. Okay, so now we're going to talk about chromosomes. We know that humans have 23 pairs of chromosomes. 22 of them, and they're numbered from 1 being the largest pair, all the way to 22 as the smallest pair. Okay, so 1 through 22 are called autosomes, and then the 23rd pair is the sex chromosome. And if there are 2 X's, then we're looking at a female. Then if it's an X and a Y, that's going to be a male. A chart that shows all of the individual's chromosomes is called a karyotype. These are 2 words that are pretty important for us. So diploid means that we have pairs of chromosomes. Haploid means they're unpaired. There's only the one individual chromosome. So diploid we write as 2N, and haploid we write as N. When a haploid egg and a haploid sperm join together in the process of fertilization, that restores the 2N number. We get 1 chromosome from the mother and 1 chromosome from the father. Alleles are different forms of a gene. So we know that a gene is the sequence of DNA that can be undergoing transcription and then translation to make a given protein. So an allele is a different variation of that gene, of that DNA sequence. Now we have some alleles that are dominant. Now, when we hear dominant, normally we think stronger and better, but that is not the case when we talk about alleles. When we talk about an allele, dominant means a trait that can cover up other forms. Okay, so on the other hand, a recessive allele is not a weaker or a bad version. A recessive allele is something that can be covered up or hidden by the dominant trait. So some keywords here that are really important for us to know. Genotype. The genotype is the genes that an individual has. What does the DNA code say for a certain trait? So a homozygous individual has 2 copies that are the same. So 1 chromosome and the other chromosome match in terms of what their allele looks like. Heterozygous individuals have different alleles for the gene. 1 chromosome has 1 version and the other chromosome has a different version of the gene. The phenotype is the trait that we can observe either in the anatomy or in the physiology as a result of the various genotypes possible. And a Punnett square is the tool that we use in order to figure out, based on the genotypes, what phenotypes the offspring might have. So we're going to look at an example of a Punnett square in the next slide. So here we're looking at the trait of a cleft chin, this little dimple that some individuals have in their chin and other individuals don't have. So a cleft chin can happen when we have a homozygous dominant genotype or a heterozygous genotype. Okay, so in a homozygous dominant, we have 2 copies of the dominant allele. In a heterozygous genotype, we have a dominant and a recessive copy, and the dominant covers up the recessive. If we have no cleft chin, we have 2 copies of the recessive trait. There's no dominant copy to cover up the recessive. So if we have 2 heterozygous parents,
Okay, so here is my heterozygous mother. She has some eggs that have a dominant copy and some eggs that have a recessive copy. 50% of her eggs will have the dominant and 50% will have the recessive. If the father is also heterozygous, 50% of his sperm will have the dominant copy and 50% of his sperm will have the recessive copy. So we have 4 different combinations that these can be combined into. We could have an offspring who inherits a dominant copy from the mother and a dominant copy from the father. They would be homozygous, dominant, and they would have a cleft chin. We could have an individual who inherits a recessive copy from the mother, but a dominant copy from the father, and they would be heterozygous. They have 1 dominant and 1 recessive, and they would have the phenotype of a cleft chin. We could have an individual who inherits the dominant from the mother and the recessive from the father, and they would be heterozygous with a cleft chin. Or in 25% of cases, we have the chance of inheriting the recessive from the mother and also the recessive from the father, in which case we have an uncleft chin. So sometimes genes carry alleles that relate to diseases, and we could have individuals who are healthy, who are carriers of a disease, or who have a disease. And there's an entire career of genetic counseling that helps people who are interested in having children or who have already had children, and advises them on the chances that their offspring will have certain genetic diseases. We do have some different patterns of inheritance other than simple dominant and recessive. So we have places where there are multiple alleles that can impact a gene. ABO blood type is an example of this. And this is specifically an example of codominance, where 2 alleles could be expressed at the same time. A and B can be expressed together in the AB blood type. We can also have incomplete dominance, where we see the offspring as an intermediate between the parents. If we want to look to plant science for an example, for instance, a purple flower and a white flower cross to make a lavender flower. We can see things similar to this in human disease as well. We have polygenic inheritance, where multiple genes, in this case 3 genes, work together to impact eye color. Or we can have pleiotropy, where 1 gene can have different forms that result in different symptoms. So in this case, the disease alkaptonuria can show up in patients in 3 different ways, depending on what specific change in the allele takes place. And we're going to wrap up with talking about sex-linked inheritance. Okay, so sex-linked traits are found on the X chromosome. So here we have a female who has 1 copy of the allele, the dominant copy, and then the recessive copy. So this person is heterozygous for the trait C. In our male, he has the recessive copy on the X chromosome, but he doesn't have that gene on the Y chromosome. It's missing. It's not part of the genes that are found on the X chromosome. So when a gene is found in the DNA of the X chromosome, we call it sex-linked or X-linked. Sometimes you'll see it that way. And its inheritance is a bit different. So colorblindness is one example of a sex or X-linked trait. Colorblindness is recessive, and the trait is found on the X chromosome. So females have 3 possibilities relative to colorblindness. First, they could have 2 copies of the dominant trait. So they would have 2 of this allele here, and if it were dominant, also a yellow dominant copy on the other X chromosome. And then they would have normal vision and would not be carriers. The case here where we have 1 dominant and 1 recessive copy, here's my dominant, here's my recessive copy, this is going to be a carrier of the trait color blindness. And finally, we could have an individual who's female who has 2 lowercase c's. So they would have the recessive trait on both of their X chromosomes. Males have only 2 possibilities. A male could have a normal copy of the color-sided trait and then no copy on the Y chromosome. And then they would have normal regular vision, or they could have the recessive trait on the X chromosome and no gene on the Y chromosome. And we see that here. We see the recessive on the X and nothing on the Y. And this individual is going to be colorblind. So I want you to take a moment, test and make sure you are understanding how you would make a Punnett square for this. This should be review from previous biology classes. Considering the trait of colorblindness, what will be the phenotypes of offspring from a normal vision male, okay, and a carrier female? So if you need a hint, here's your normal vision male, and here is your carrier female. So right now, I'm going to recommend you pause the video, get out a sheet of paper, get out a pencil, and actually draw a Punnett square and see if you can do it. Don't just think about it because you'll think wrong. I guarantee it. I've taught this stuff for a long time. Get out pencil and paper and draw it. Once you have your answer, then play the video. So, I sincerely hope that you gave yourself the chance to practice this and you have something on a sheet of paper in front of you. You should have put the mother with 50% of her eggs having the normal allele and 50% of the eggs having the colorblind allele. Half of the sperm for the male should have the normal vision traits, and the other half of sperm should have no gene for vision. So we have 2 ways that female offspring could be born. Okay, so the females could be completely normal vision, non-carriers, or the females could be carriers. And then we have 2 different possibilities for the males. We could have normal vision males, or we could have a male who is colorblind. Okay, so some things that I want you guys to recognize in this. Both of the parents have normal vision. The mother has normal vision because she has the dominant normal vision gene, and the father also has normal vision. Yet we have 25% of the offspring who are colorblind. We can ask lots of questions about this, and you might expect to see some questions on an exam. So if I were to ask, what % of daughters are colorblind? I hope you just said the answer is 0%. If I asked you what % of males are carriers, I hope you told me 0%. Males can't be carriers of sex-linked traits. If I asked you what % of offspring overall will be colorblind, I hope you just said 25%. If I asked you what % of males will be colorblind, I'm hoping you just said 50%. So I hope all that made sense. If it didn't, please send me an email so we can clarify anything that you're confused about. All right, some final thoughts on alleles. We already talked about how dominant alleles aren't necessarily better. They also aren't necessarily most common in the gene pool. Okay, so type O blood is a recessive allele, and in the United States, it is the most common blood type. We also have dominant alleles such as AB and polydactyly. Polydactyly is the gene that causes someone to have 6 fingers or 6 toes on a hand. That is also dominant, but we see it very rarely in the population. So now we're going to talk about something a bit different. Epigenetics is a way that gene expression can be changed. So we can see different patterns of gene expression. Epigenetics regulates gene expression at the DNA level by turning on and turning off some genes with a process of methylation, which is adding methyl groups to sections of DNA to make them easier or more difficult to replicate. And we're going to wrap up by talking about cancer. So when we talk about cancer tumors, malignant tumors, or tumors that metastasize, we're talking about a tumor that has the ability to break off and travel to new places and cause new tumors. That's what a malignant tumor is. Oncology is the field of medicine that studies cancer and cancer tumors. We have several different types of cancer, and they are defined here. Carcinogens are things that we can encounter in the environment that cause cancer. Radiation, chemicals, and viruses are 3. Cancer does have a genetic component, and a small percentage of cancers can run in families. 2 different types of genes are related to regulation of cancer. So oncogenes cause cancer to grow, but tumor suppressor genes stop cancers from growing. So these are things that we consider when we look at a person's genome. And finally, cancer is lethal because it can invade the blood vessels or other critical tissues such as lung or brain tissue and cause damage to those things that's detrimental to the whole body. Cancer cells have very, very fast metabolism. We talked about this way back with PET scans, where we were looking at the cells with high metabolism, and cancer cells show up in those types of scans. So cancers take nutrients away from the rest of the body. They also allow other infections to come in because the cancer patient's immune system is weakened. So this picture here is something called Willis's cancer or Willis tumor. It's found in kidneys, and it's specifically a pediatric or childhood cancer.